don't think it matters.
Okay, let's see if number one.
Maybe? If it works.
And I chose the wrong one. There you go. Good morning, everyone. My name is
Please fix the
Tiago,
timer.
and oh, the timer. 4 minutes.
No. Please fix the timer for the speaker. These are actually extra 4 minutes that Simon has.
We can keep the extra 4 minutes. That's fine. Okay, I'll just get ahead and start. So, hi everyone, good morning. First of all, thank you so much for having us. This is actually I'm one of the new faces that Dave was earlier talking about, so it's a pleasure to be here today. Yeah, this is just a quick disclosure, and I just want to say, I know that there is some expectation about what we bring on today. We just had our last patient last visit last month, so we still don't have any results to present. We will have these in July. But we still wanted to be here to provide some update on the status of the study, really, and there are some new information that we also wanted to share about it. So, yeah, that's me. I'm Tiago, and really we're just, as I was saying, just provide a brief update on the trial and the overall program of BIA28 or Parisiract. So I'm not going to spend so much time here, and the previous talk just pretty much did this part of the job, thank you. BI28 is a novel allosteric activator of GCase that is being studied as a potential treatment for GBA PD. And the Activate trial is a phase 2b study that is aiming to evaluate its efficacy and safety in terms also of delaying clinically meaningful progression in PD. This is a sort of the overall design, which you probably already know. I should say one thing, we ended up enrolling more than 237 patients. We actually randomized 272 patients. This was not initially foreseen, but at the time that we enrolled the 237 patients, we had already a few patients that had already signed informed consent and all of that, so we didn't really feel that it was ethical to just say, look, you've already consented to participating in the study, but you're not actually going to be able to. So as long as they complied with inclusion criteria, we let them in the study as well, and this led to a final number of randomized patients that was above the initially planned one. And this is just an overall scheme of the design. We had an initial period of screening, genetic screening, but also Parkinson's disease criteria and many other criteria, and then they went on to do a 78-week treatment period plus a safety follow-up. We tested 2 doses, the 10 and the 60 milligrams, and the third arm, the placebo arm. And we did this across many countries, here in the US, as well as Canada, and then a few other European countries. Our primary endpoint and key secondary endpoints, these are time to event endpoints, and we're talking about the MDS-UPDRS parts 2 and 3. But also looking at other scales, also looking at these scales from a change of baseline perspective. We've also incorporated a digital wearable study, as well as an optional imaging study, which I'll also provide some inputs on. And then obviously the biomarkers, which are key to this program, namely GCase activity, substrates, also some other markers of or potential markers of neurodegeneration, namely NFL, and other markers of lysosomal activity as well. So this is really just a summary of the kind of patients that we included, between 1 and 7 years of diagnosis. These are by themselves patients that are not super easy to find, so we didn't want to really, narrow the inclusion criteria too much to the point that it would just be a challenge or pose a challenge in terms of recruitment. Patients were allowed to stay on their symptomatic therapy as long as this was a regimen that had been stable for at least 30 days before the initiation of the study, and then, modified autoimmune motor complications, as you can see in the screen. We try to target early to mild PD patients, and obviously the presence of a GBA-PD associated variant. This is just a list of the mutations that we included. If a patient presented with a mutation that was not included in the list, but that was proven to be increasing the risk for Parkinson's disease, we would allow these patients to get in the study as well. And in terms of our initial numbers, so we had initially expected to have to screen, around 3,700 patients to get to our enrollment target of 237 patients. But we ended up screening less than a third of that. And if we look at our screen patients and the GBA positive patients, we're looking at a ratio of around 31%, which we know is not in line with the reported prevalences of these patients in general. But this, in fact, reflects just a very strong, feasibility assessment right at the beginning of the study, engaging with key investigators in the area of GBAPD, engaging with investigators that have long-standing experience in trials, engaging investigators that have, sort of a database of patients that were already pre-identified in terms of their genetic status. Obviously, for purposes of the study, they had to repeat or reconfirm their genetic status, but some of these patients actually already knew that they were GBA positive. A regular engagement with, for example, the Parkinson study group here in the US, but also NSPARC in France, and really targeting countries that have a well-structured and robust GBAPD database. We ended up, as I've said, enrolling 272 patients. So just a quick glance at our numbers. It took us around a year and a half to randomize these 272 patients. As I've mentioned, we've just had our last patient last visit last month, and we are currently in the process of looking at all the data, database lock, all of that. So we're targeting July 26 to have our top-line results going public. So we're very excited for that, and yeah, please stay tuned for that as well. And just some general characteristics, we ended up enrolling the most patients here in the US, followed by Italy and Spain. The US actually started the recruitment 6 months earlier, and keeping the European investigators on par with the numbers that we were achieving in terms of enrollment in the US actually sort of motivated them to also, try to be on par. And at the end, we ended up enrolling 107 patients in the US and the remaining 165 patients in Europe. And of course, 6 patients in Canada. So most of these patients were men with an average age of 58 years. And while I can't disclose yet the specific variants that were included in the study, what I can say is that the frequencies of the variants that we found in the patients that were enrolled are actually very close, very similar to the reported prevalences of these mutations in PD patients in the literature. And we ended up enrolling about a third of patients with a risk variant, a third of patients with a mild GBAPD mutation, and a third of patients with severe GBAPD variants. In terms of our numbers, so as I've mentioned, we've enrolled 272 patients. We've managed to finish the 78-week treatment duration and safety follow-up with 230 patients, which for us it's a huge achievement. And we managed to keep our early termination rate at 15%, which we also feel that for a 78-week trial for disease-modifying therapy that yields no immediate symptomatic benefit, this is a great achievement for us. If you look at our reasons for discontinuations, most of them are withdrawal of consent, and I'll get into that in a second because this relates very strongly with our patient retention strategies that we had to adapt as the trial was ongoing. Some adverse events, but I should say that more than 80% of these were not related, so we didn't really see any safety trends in this trial or with the molecule, so we're also quite happy with that. In terms of our digital wearables sub-study that I mentioned before, we've engaged with a lot of key opinion leaders working in PD, but also in these digital assessments. And we were constantly getting the feedback that if you want to do this, implement this in your trial, you should aim for measures that are tangible for a clinician that is actually seeing a patient. And we're talking about measures like gait speed, we're talking about measures like turn speed, tremor, bradykinesia, and we were always getting the feedback that, in these sort of trials, we're looking at maybe a completion rate of 60 to 70%. So the fact that we've actually managed to get around 80 to 90% of patients completing all the digital assessments that, were done throughout the whole study is actually a very satisfactory number for us as well. For our sub-study of our imaging sub-study, so we focused on arterial spin labeling and diffusion tensor imaging with a focus on, free water imaging in the substantia nigra and other regions, and also just, basic MRI volumetry. We've managed to, so this was basically a 2 MRI sort of sub-study, one at the beginning before treatment and one at the end, whether it was week 78 or at the moment that the patients would early terminate. And we've managed to complete with 52 patients. These were not really stratified in terms of treatment. This was an optional sub-study. So we still don't know if, all of these patients are placebo or all these patients are active. We're hoping that we will get a sort of a balanced allocation, but yeah, we can't say that for sure for now. But it will be interesting to also have this imaging data and integrate that within, biomarkers, digital, and of course the clinical assessments. I wanted to leave some part of the discussion to address some of our challenges throughout this trial, because it's not easy to conduct a 78-week trial in Parkinson's patients. It's not easy to find, these patients with GBA variants. And in fact, one of the things that I've mentioned earlier, one of the main challenges started right from the beginning in terms of finding these patients and going to the right investigators, selecting countries with very well-structured GBA PD databases, or PD databases for that matter. The adjustments of anti-PD medications, I guess this is something transversal to any program in Parkinson's disease, not just GBA, but in fact, we're talking about a 7-8 week trial, a year and a half. This is very long for a Parkinson's patient. And one of the things that we learned very early on in the trial was that we had to have very strong sort of, patient education initiatives, because and we saw this with our patients in this trial, when we saw that we started getting very frequent feedback from patients that, there is this expectation, they're getting into a trial, there is this expectation that they will feel some sort of benefit or immediate benefit, but we're not talking about a disease-modifying trial that is not expected to yield any immediate symptomatic benefit. This leads patients to question very often, is this thing working? Should I, just commit to this 78-week trial study? Because these are heavy studies, collecting all the clinical assessments every 13 weeks, doing the digital assessments, coming in for their MRIs. This is a lot for a patient. And we had to, try to transmit to the investigators and to everyone involved in the study to try to, transmit this message to the patients that, this is how we hypothesize that the drug will work. So even though you might not feel some immediate benefit at the moment, what is expected is that at the end, maybe you won't be as worse as someone who did not, or who was not treated with the drug. And in fact, these adjustments of anti-PD medications throughout the trial, because throughout a year and a half they will happen, are something that we also had to tackle from very early on in terms of how can we make sure or try to ensure that this will not affect our analysis, and obviously that pertains to the type of analysis or type of primary and key secondary endpoints that we've chosen, and really about the patient withdrawal, the 15.4%, which is still for us a very satisfactory percentage, but we've had to engage with the patients very often. We were distributing newsletters periodically to provide a status update to keep them engaged with the study. And yeah, so obviously there are many challenges in such a big trial, multi-center trial. But when I was thinking about this, and if I had to highlight at least three, these I guess would be the three main challenges that we have faced throughout these last 3 years. And just a few final notes. So the the program is moving forward. We're keeping our engagement with our authorities. In fact, we've just had earlier this year an interaction with the FDA where we've discussed many topics. It was a meeting with the FDA where we've discussed topics, around obviously our statistical analysis plan, clinical pharmacology studies, but also about the whole question of disease modification, about endpoints, about minimal clinically important differences, which I guess are some of the questions that maybe almost everyone working in the PD field has at this point, because there are no clearly defined thresholds for some of these scales, and maybe for one authority, one thing is clinically meaningful. For an European authority, we might get a different opinion. And in fact, we had dissonant opinions in terms of what is clinically meaningful for these patients. So trying to integrate all of that is not easy, and actually has propelled us to keep this ongoing engagement with them. So as I've mentioned, our results, they will be available in July 26, the results of our 230 patients. We've also have some additional, just phase 1 studies, clinical pharmacology, renal impairment studies, and so on ongoing. Some of these results will be shared in upcoming conferences, so stay tuned for that. And yeah, this was just a brief overview of that we wanted to share with you. I sort of, skipped a bit through the design of the study, but obviously if anyone has any questions about that, we are more than happy to take them. And this was actually my last slide. We're still 4 minutes ahead of time, so that gives us an extra 4 minutes for questions for both me and Simon. And yeah, thank you so much for your attention.